A+ലേക്കുള്ള വഴി ഇനി തനിയെ തുറക്കാം. Subscribe to the ENCLAPS channel. Ring the bell button and open success. It's that easy.
Welcome all to the wonders of the living world. First, let's watch a small video. What have you seen? A boy eating a bread, right? Do you like toasted bread with jam and omelette? Well, I do. But have you ever thought of how a bread is baked? Well, today we are going to learn the technology that helps us to understand how our life is easier. Though the process behind it is sophisticated, the idea and the end results have proven to be beneficial for mankind. We have learned that genes, to be specific, DNA, are the ones that create differences in mankind. Each person differs from other because of the genetic variation. But still then, there are many genes that are common within us. Now let's start the chapter with a term called genetic engineering. Have you heard of this term earlier? If not, let me explain. Genetic engineering is the direct manipulation and modification of an organism's gene using biotechnology. Hey, another term called biotechnology. What does that mean? Biotechnology is the technology that utilizes biological systems, living organisms, or part of them to develop or create different products. Now let me explain this technique using an example.
What is this? Tomato. You might be wondering if it is potato or tomato. It actually is both. Potato plus tomato. In this plant, tomatoes grow above the plant and potatoes grow below the plant. It can be done by a process called grafting. In this, both the stems of the plants are joined by tying them. This is an example of plant engineering. Similarly, when we cut and join genetic material, the process is known as genetic engineering. It is the most important principle of biotechnology. Most of the principle of biotechnology is the base of genetic engineering. Hence, genetic engineering deals with the modification of genetic material of an organism. There are mainly two types of genetic material. DNA, which is double-stranded, forms the genetic material of most of the organisms, and RNA, which is single-stranded, forms the genetic material of many viruses. Hence, genetic engineering deals with the manipulation or modification of genetic material of an organism.
I hope you've got an idea of what is genetic engineering and biotechnology. Do you remember in the beginning I have given another example of baking bread? The process too falls under the concept of biotechnology, the use of yeast, a living organism, to produce the desired product. Such traditional processes usually utilize the living organisms in their natural form or further developed by breeding, while the more modern form of biotechnology will generally involve a more advanced modification of the biological system or organisms. Now listen to the doctor's talk conducted by the health club. In between the talk, Saju came up with a doubt. Now let's hear what is Saju's suspicion.
How can the insulin produced by bacterium be used by humans?
That's a very good question. Have you thought about it? How can bacteria produce insulin that can be used by humans? Remember, we started with the term genetic engineering. So this technology can be used for the production of insulin from bacteria. Now let's understand the process. The various stages in the production of bacteria that are capable of producing insulin. Step number 1. Cut the gene responsible for the production of insulin from the human cells using restriction enzymes. Second, join insulin producing gene with the plasmid. Plasmid is a circular DNA of the bacteria using the enzyme ligase. The gene responsible for the production of insulin becomes a part of the bacterial DNA. Plasmid with the ligated insulin gene is inserted into the bacterial cell. Bacteria that can multiply in the bacterial culture produce inactive insulin. Inactive insulin is converted into an active form and utilized. Now see if you can explain these indicators. What is the change that has occurred in the genetic constitution of the bacteria that produce insulin? We can cut bacterial DNA, that is plasmid, and the gene responsible for the production of insulin in humans using restriction enzymes. Then join the gene responsible for the production of insulin in humans in bacterial DNA. Thus, the genetic constitution of the bacteria gets changed. How do the new gene become a part of the genetic constitution of the target genes? The DNA from the target genes enter the target cell. Thus, the new genes become a part of the genetic constitution of the target cell. The use of microorganisms and biological processes for various human requisites is called biotechnology. From BC 4000 onwards, organisms like yeast, a kind of fungus, were used to prepare food like cake. The ability of the fungi and bacteria to convert sugar into alcohol was utilized to make wine and appam. These can be considered as traditional methods of biotechnology. Genetic engineering is the modern form of biotechnology. So we can again define genetic engineering as the technology of controlling traits of organisms by bringing about desirable changes in the genetic constitution of the organism. The basis of this technology led to the discovery of the fact that genes can be cut and joined. Now, how are the very minute genes cut and joined? Enzymes are used to cut and join genes. The main enzymes include restriction endonuclease, which is used to cut the genes. So, this enzyme is called genetic scissors or molecular scissors. Now, another enzyme called ligase is used for the joining of the DNA fragments, and this enzyme is called genetic glue or molecular glue. Now, how was the insulin producing gene of humans transferred into bacteria? A gene from one cell is transferred to another by using suitable vectors. Vectors that contain the ligated genes enter the target cell. Example, plasmids. Plasmid is present in bacteria, is an example of a vector. Thus, vectors with the required gene, we call it as the gene of interest, is called rDNA or recombinant DNA. That's all for the day. See you again with more knowledge on genes and their products soon. Till then, bye.
3 ലക്ഷത്തിലേറെ വരുന്ന ചോദ്യോത്തരങ്ങൾ ഏറ്റവും കുറഞ്ഞ ചിലവിൽ സ്വന്തമാക്കുവാൻ ഉടൻ തന്നെ ഡൗൺലോഡ് ചെയ്യൂ എൻക്ലാപ്സ് ദ ലേണിംഗ് ആപ്പ്.